Okay, we're back, and now we want to look more specifically at the characteristics that describe each of the three domains, and we'll start with bacteria and archaea. And that's mostly what we'll be doing from now on, is looking at the groupings of organisms. We need to understand the names of those organisms and their groups, and we're going to understand what characteristics put them in that grouping. And we'll start to look at, with bacteria and archaea, we'll just focus on kingdom and phyla because they are a little bit harder to classify. But as we get into eukarya, and especially animalia, we're going to look closer at the rest of the groupings and get more specific. Okay, so again, here's a nice phylogenetic tree showing the three domains. And we're starting off with the bacteria. They're classifying these as different phyla. They refer to them as phyla at this point. They haven't put them in kingdoms. And again, because some organisms are a little bit harder to put into the classification systems that we have now. And we're also just now developing better and better technologies to be able to study and understand single-celled organisms as small as bacteria. They can get pretty small. Right? But as more genetic studies are being done, you can see a lot of this horizontal gene transfer as well as the line of descent that we're understanding for them. Don't worry, we're not going to go over all of these. I've only picked out a few. Archaea, you notice by the key, they currently have 4 kingdoms. And let's start with the domain Archaea. Right? If I can click this, there we go. So in the domain Archaea, like I mentioned, they currently are divided into 4 kingdoms. There are some researchers now that would like to collapse those into 2 kingdoms, the 2 top that you see there, Crenarchaeota and Euryarchaeota. But your book went with the 4, so I went with the 4 here. And again, depending on what source you read, some will call them kingdoms, some will be a little more loose and not want to classify them into the grouping of kingdoms. Again, it's because single-celled organisms can be difficult to understand, and we're still learning about them. Okay, so here are the characteristics of the 4 kingdoms of the domain Archaea. So the Crenarchaeota, these are classified as, they're all extreme thermophiles. If you remember, that means that they can tolerate very, very high extreme heat. Okay, they're also known as the main contributors to carbon fixation. Okay, some of them are in the ocean and they float around as plankton, and they're generally found as in cold, oxygen-rich seas. And few of them are photoheterotrophs. So if you remember that term, a photoheterotroph means they use photosynthesis to create some organic compounds, but they do also have to eat and gain organic compounds from the environments. That's the heterotroph part. Okay, so those are the 4 main characteristics describing Crenarchaeota. Okay, Euryarchaeota, these are the group of archaea that are referred to as methanogens. So they end up producing methane gas from simple carbon compounds. In order for them to do that, they are obligate anaerobes. Remember, something that is anaerobic cannot use oxygen. It doesn't use oxygen to create energy. So they don't want to be around oxygen. And especially if they're obligate anaerobes, then that means they can't be around oxygen. Oxygen actually poisons them. So they're found in areas of the earth, like deep in hydrothermal vents and volcanoes and things where there's no oxygen available. Some of them are also known as extreme halophiles. Remember, halophiles refer to salt. So they're heterotrophic and they need large amounts of sodium in order to grow. Okay? So that's the Euryarchaeota. The Corarchaeota, these share characteristics that are intermediate of Crenarchaeota and Euryarchaeota. They may have branched before Crenarchaeota and Euryarchaeota separated. So again, that's one reason why you might see core Archaeota in a different grouping, but this is how yours grouped it. They've been found in terrestrial hot springs. So this is the only characteristic I'll put here for core Archaeota that you have to remember. And then the last one, nanoarchaeota. Nanoarchaeota, nano means small, and they're very tiny, and they have a very small genome. And they also live endosymbiotically attached to another archaeon. Okay, so that's as deep as we're going into the domain Archaea. We still don't know a lot about these single-celled organisms, so I'm going to just introduce them to you this way. All right, the domain bacteria, we studied them a little bit more when we looked at single-celled organisms, right? There are, when you look at the cladogram before, there were several different phyla. I'm going to just pull out 5 major groups because they're the ones that are the most widely distributed in our environment. So the first group is the proteobacteria. These are classified as gram-negative bacteria. You might want to go back to the earlier lecture in the semester to understand what that is. But there are 3 types that I just want you to remember. So there's the alpha proteobacteria, and these are important because they're symbionts of plants and animals. Some become pathogens, but remember the symbionts of plants and animals, it's the alpha proteobacteria that during an endosymbiotic event became the chloroplasts in plants and became the mitochondria in both plants and animals, or I should just say eukaryotic cells, right? So that's one of the reasons why alpha proteobacteria are pretty important to remember. Again, some of them can cause sickness in us. The gamma proteobacteria, these are really important decomposers. So they break down decaying plant materials and things. So that's very important, we saw in our ecology. The other one type of gamma proteobacteria is one that inhabits our intestinal tract. So that's Escherichia coli, or sometimes we just call it E. coli, right? So from our earlier lesson on binomial naming of species, the genus Escherichia, this is actually after the name of the researcher that discovered it. I think his name was Escher, I believe is how you pronounce it. And then the genus name is Escherichia. I can't say it that well. So that's the noun, but that's named after a person. Coli refers to where it's found. So that refers to the colon. So it's found in the colon, right? So very important, remember, we don't survive well without E. coli. Some can cause harm to us, but in the right balance, our E. coli helps us in our digestive tract. And then epsilon proteobacteria, also found in the gut, but usually in the stomach. And the species is Helicobacter. This is the genus name, and it can cause peptic ulcers. Right? The next grouping are the cyanobacteria, also gram-negative, and I put this in here because remember they contain chlorophyll for photosynthesis. And when the cyanobacteria evolved, these were the single-celled organisms that transformed the atmosphere. So because they started taking water and carbon dioxide, the waste product is oxygen, and they release O2 into the atmosphere. So that's what released free oxygen into the atmosphere, completely transforming our Earth. Okay. The other 3 groups here, we've got chlamydias, which are gram-negative, spirochetes, and also gram-positive bacteria as well. Let me hang on a second. Oh, I can't. Okay, sorry about that. I had a little blip. I was getting interrupted and I wanted to make sure I could deal with that. So we talked about cyanobacteria. The next one are the chlamydias. Chlamydias are energy parasites, and so what that means is that they are completely dependent on their host for ATP. right? So they can't make their own ATP. They infect almost every species of bird and mammal. And so it's also in humans a major cause of pelvic inflammatory disease. So it's a sexually transmitted disease that can occur there. The spirochetes are a spiral-shaped bacteria. They have these interesting movement with flagella. They can be disease-causing. They actually produce syphilis in humans. And then the gram-positive bacteria is a really large group of bacteria, and some of the main ones, they can cause disease in people. So things like streptococci, they form strep throat, right? So they get in the mouth and the digestive tract of humans, and again, causing strep throat. There's staphylococci. So if you get a staph infection, that's from staphylococci. And there's many more, but I didn't want to go too in depth into those guys. So if you just remember a few of the organisms that are related to humans, then you're good to go. All right, let's get into the domain Eukarya. So in the domain Eukarya, the first kingdom that we can talk about is the kingdom Protista. So again, remember we looked at these already when we were looking at the form and function of organisms, and we had to start with single-celled organisms within the Eukarya, and that started with the kingdom Protista. And so again, this is one of those paraphyletic groups that now it moved from polyphyletic to paraphyletic because we're learning a little bit more. So it's paraphyletic now because we're still kind of basing on past attempts to classify them. But so what they've done is by using molecular data and something called ultrastructure, that helps to establish a different way of grouping the Eukarya, and it helps to really focus on Protista, but the other groups, other kingdoms will be involved in this. These are called supergroups. So this would be outside of the Linnaeus classification. You can think of supergroups as in between domain and kingdom, okay? Whereas the Protists, though, really hard to classify them within separate kingdoms. So they're just calling them supergroups for now. So again, they're based on new technological data. It's called ultrastructure, and that means it's the really fine detail of cell structure. Oh, actually, I had that on the previous slide here. I can go back there. All right, so this is what ultrastructure is. Using electron microscopes so that you can see the real fine, it reveals the fine structure of each of these. These are all single cells, things you couldn't see with the naked eye. And so it's almost a way to anatomically group organisms together, the protists together. If you remember this guy, Diplomidad, we saw that one. This one I think is a diatom. So that helps. And then also using ribosomal RNA, right? When you sequence ribosomal RNA, we're starting to see commonalities between different groups. So that's what gives us the these 5 supergroups, right? So the excavates, the chromaviolates, the rhizarians, the archaeoplastids, and the unicons. So again, this arrangement kind of shows the importance of protists in understanding our evolution. Okay, so this is the real simple, just to show you the 5, that we all came from an ancestral eukaryote. But in the next picture here, this is a little more detailed phylogenetic tree showing all the supergroups. And so again, this is mainly shows you, again, the importance of the protists. And so many supergroups only contain protists, like in the excavates, the chromaviolates, and the rhizarians. But look, then you get to archaeoplastids, where you have protists, the red algae and the green algae, but it also includes land plants. So this arrangement of supergroups now is showing us the evolutionary pathway from protists to plants, to a complex organism like plants. And the same with the next one, the uniconts, right? The uniconts are also mainly the protists, but we've got two kingdoms in here, the fungi and the animalia. And so again, this helps us understand that evolutionary relationship between the protist groups and the animals as well as the fungi. Okay, so again, if they're in the oval right there, then those are organisms within separate eukaryotic kingdoms. So all the other organisms, all the other eukaryotes in this tree are informally known as the protists. Okay, and you still see we call it the kingdom Protista, but they're more and more just being separated into these protist groups. Okay, and just a little more. So again, just to read this properly. So again, node A, it shows that all 5 supergroups branch from the same point. And this just means that it's unresolved at when those would branch. So I said usually at the levels of branches shows you the timing of the evolution. So this doesn't mean that there was an ancestral eukaryote and then bam, 5 groups branched off of that. We just don't know what that is yet. So at node A, they just have the same one. Okay. Let me see. The same is true at node B. So we don't know the timing of the evolution of diatoms, brown algae, and golden algae, right? We know they're related within the chromaviolates, but we just don't know the timing on that yet. Okay, so if you wanted to know where is the common ancestor of brown algae and water molds, well, here's our brown algae, right? And water molds are way over here. So you just kind of follow their lines until they cross. And so that would be here. This would be the node, the common ancestor of brown algae and water molds. Okay. What about animals and fungi, right? Same thing here. Here you've got animalia on this line, fungi on this line. Finally, where they cross, that would be the common ancestor of fungi and animals. And so what this suggests is that there was a common ancestor of fungi branched off, and then the line continued, and then the choanoflagellates branched off and the animalia branched off, right? So fungi probably a little more ancient than animals and choanoflagellates. All right. So again, what you need to do, you need to know all 5 supergroups. And you need to know the representative protist clades and then the key characters of each one of those, right? And so you have this table in your book, table 26.1. You can use it on the PowerPoint, however you want to do it. And so I'm going to just kind of step you through each one of these real quickly through the rest of this PowerPoint. But this is what I'll ask you. What is the supergroup? What is the representative protist clade? And what are the, maybe name 1 or 2 or 3 key characters of that group is what I could ask you to do. Okay, and then where it's appropriate in the Archaea, Plastids, and the unicont, I might ask you about how land plants and how animalia and fungi fit into those. Alrighty, so let's go through each one. We'll start with the excavates at the top. And so if you might remember some of these pictures, we look at the excavates when we looked at protists, single-celled organisms. So this is the first group. These are unicellular protists. Remember, they have really modified mitochondria. They're considered what are called biconts. That means they have 2 flagella. And in fact, all of the supergroups have 2 flagella except for one, the uniconts, right? So the representative protist clades that fit into the excavates, remember the diplomonads and the parabasalids, also the euglenoids and the trypanosomes. Remember euglena, these were, they can photosynthesize. If they're not in the sun, they can lose their chlorophyll and they can become heterotrophic. And remember the trypanosomes can cause that disease African sleeping sickness. So key characters of the excavates, 2 or more flagella. They have a ventral oral feeding groove. You can see that here in the what's it called? What's this guy called? The euglenoid, the euglena, right? They didn't highlight it here. They didn't label it, but there's a feeding groove there. And then crystalline rod in the flagella. It makes their flagellum a little bit different than others. Okay, so those are the excavates. The next supergroup are the chromaviolets. These are really diverse. There's a lot of protists that we put into the chromaviolets. They're probably originated as a result of secondary endosymbiosis, and it's probably ancestral cell engulfed in red algae, right? And so these are also considered biconts, and so they are characterized, remember the dinoflagellates? They actually have 2 flagella in that groove in the middle. And dinoflagellates, remember, when they bloom, reproduce by a lot, they can produce that red tide in the water. Okay, so there's dinoflagellates. There are also the ciliates, remember, like paramecium, so they have cilia all around their outer edge. Okay, the epicomplexans as well. I didn't put an epicomplexan up here, but I did show you the water mold, also diatoms as well, and the two distinct types of algae, the brown algae, which can be multicellular. So this is the macrocystis, this is kelp, right? So that's brown algae, and then also the single cell golden algae, right? So again, what they have are alveoli, which are flattened vesicles just inside of the cell membrane. You can see a little bit in the golden algae here. Those are those vesicles. Sometimes, yeah, this one isn't powerful enough. It's in the cell membrane. Sometimes you can see it on the paramecium, but this isn't zoomed in enough to see that. And then again, most have 2 flagella, and at some point they may have had the flagella, but they lost it over time. And so, yeah, those are the key characteristics of the chromaviolets. Okay. The next one is the rhizarians. Rhizarians are amoeboid cells that have tests or shells, right? So that's like the actinopods. See these little rays you see sticking out? That's actually part of the cell being pushed outward, but they do have a shell around the outside. That's the real kind of the clear material that you see there. So we call them amoeboid because they're not like the amoebas we'll see later. So the difference is the shell or the test is what that's referred to, right? And so this one is showing foraminifera, also known as forams, where you can see the test really well. It's not clear. It's not as clear. So again, a porous test known as a hard shell where the pseudopods can extend through. Okay. And they might even have an internal shell referred to as an endoskeleton. Okay, so forams and actinopods are part of the rhizarians. Okay, next group, next supergroup are the archaeoplastids. These are the red algae and the green algae, right? So they have plastids. They're bound by an outer and an inner membrane. These are the chloroplasts, is what that means, an organelle called the chloroplast. And it has a pigment in there that would be to help absorb the radiation from the sun to produce energy. So the red algae, they have different pigment if we classify them as red algae or green algae. Okay. Red algae, you don't need to know this, but I'll put that in there. It's a phytocerathin, which is a red pigment. That's what gives them the red color. And the phyocyanin is the blue pigment, but it really gives them a green color. So that's the green algae. And so again, the different pigments help them to absorb different wavelengths from the sun's radiation for photosynthesis. Okay, so here's the green algae, and then remember that also these are, we're talking about the protist groups in there, but what also belong in the archaeoplastids are the land plants. So remember that land plants share a common ancestor with the green algae. Okay, and especially the stonewort, this plant is called the stonewort, is very closely related to land plants, and it's widely distributed in fresh water. So if you remember, these being in fresh water, probably along the shoreline, are probably the groups then that diverged and became the land plants as they tolerated more dryness. Okay, the last group are the uniconts. So the uniconts are cells that have a single flagellum, right? Therefore the term uni, meaning one, unicont. Okay, or it does also include the amoeba, and they don't have any flagella, right? But genetics suggests that their common ancestor, they evolved from a common ancestor with a single flagellum, right? This group also includes the animals and the fungi, but for the other single-celled organisms, you've got the amoeba. So again, these are the true amoeba. It also includes, remember, slime molds. I think they're so cool, slime molds, and then also includes the choanoflagellates, and it's the choanoflagellates then that share the common ancestors with Animalia. Alrighty, so again, make sure you know the supergroup, the name of it, a representative protist, but you should also know where plants, fungi, and animals fall in. And then some of the key characters. And I also put, this is from your table, just another way of showing it. So if this is easier to print or something, you can print this off. That shows all of that. And I think the study guide asks you to do something like this as well. All righty, that is it for systematics and the single-celled organisms. Next, we're going to start to get into multicellular organisms. All right, thank you, everybody.